Welcome, everybody. I can't see anything. I guess it's just you guys can see me. So hopefully the day's going well. So today I'm going to talk a little bit about the immune system and how it links to behavior and brain development. And this is part of what we study in the immune platform, which I'm sure everybody's aware of, within the PON network, which is now co-led by myself and Chao Wang, who's an assistant professor at the University of Toronto, whose lab is at Sunnybrook also. So immune brain communication, it's important to remember that when we're talking about immune brain communication, we're not talking about inflammation per se. So a lot of times when people think about the immune system, they think about infections or, what happens when you get an infection or pathogens. But we need to start first with the fact that the immune and the brain are in continual communication. The immune system communicates with the brain. It gives them some signals of what's happening in the periphery, but also the brain sends these nerves throughout the autonomic nervous system to control all aspects of your body, including the immune system. And so there are many ways that your body can talk to your brain through neural pathways, through messages in your blood, or through actually immune cells, which we know a lot more about these days, that particularly during early development are primed by body sites like the gut, and they actually go and travel and they live in the meninges, which is that outer area of your brain. And by living there, they actually release chemicals or molecules that influence all sorts of aspects of brain development. And in the long run, they can influence behavior. And so when we talk about the immune system and we talk about the brain, what we know now, and we've known for about 20 to 25 years, is there's a lot of molecules that are actually signaling in the brain that are similar to those that are operating in your peripheral immune system. And so there's a couple of areas that we're most interested in when it comes to the biology that might help us understand why some clinical presentations across neurodevelopmental disorders might be different. And if we start it from the top and work our way down, we know that within the brain, there's neurons, there's astrocytes, there's glial cells, but one of the key cells that is important to immune brain communication is the microglia. And so the microglia is, in fact, the innate immune cell in the brain. It comes from the yolk sac very early in development, and it can respond within the brain to things that are happening. But one of the things that's very important to is it responds to peripheral cues and influences how neurons connect during development. And one of the ways that it does that is it helps the nervous system decide which of those connections are the strongest and which need to be removed. So it actually is important for synaptic pruning and some of those events. And so, like all of the immune system, the microglia cell is there to keep things in homeostasis. But in general, what we think about the immune system is it's there as, first of all, a first responder if something's happening, but also its job is sort of to dampen down whatever that reactive situation is and make it back to this sort of good homeostatic balance. And if we look out in the blood or in the tissues, we can see we have a whole host of different immune cells, such as your lymphocytes or your monocytes. But these T lymphocytes or these monocytes can both traffic around the brain, but the other thing they do is they can produce these immune molecules that can actually signal to the barrier cells of the brain and link to a cascade of reactions that can change things within the brain. And this is what happens when we have an infection. And that's one of the reasons why we sort of get sleepy is part of that action are these cytokines that actually trigger a response in your brain that tell your brain in an adaptive way to sort of rest so that you can recover. And then finally, the last level of communication that is of interest in both my lab, and it has been an emerging interest more and more over the last decade, is how these trillions of microbes that are in your gastrointestinal tract influence the immune system. And so changes in your microbiome or your gut bacteria in response to diet, to lifestyle choices, to your own DNA can communicate with the immune system that is in that mucosal part of your gut, and then have a pathway to communicate with the brain. So these are all pathways that are present during normal, healthy day-to-day life. And they're also the pathways that sometimes get disturbed that might be contributing to symptoms of diseases. And so I have a couple of summary slides here just to remind myself to make sure I touch on all the different things that are important. And so these gut bacteria are certainly of interest, and they cover all the surfaces of your body. They don't just include the bacteria, although the bacteria is the best studied. They also include viruses and fungi, protozoa, and parasites. There's trillions of viable bacteria in your gut, and they're essential to the development of your immune system. And this is why both the gut and the immune system can't really be looked at independently. And then there's this bidirectional communication that influences brain development. So the peripheral immune system communicates with the brain, as I said, through these neural pathways, through humoral pathways, or by cellular pathways. And it can, in fact, activation of your immune system in the periphery can actually activate food hormones and other hormones. So it's not just these simple things. It can also change the way that your body metabolizes food, or the body's host metabolism as well. And those change in metabolites is something that we can measure either in biospecimens such as the blood or in urine. And they also help us map what some of the signaling pathways are between your body and your brain. Your circulating immune system influences normal brain development. That's one of the themes. But then if we think about it, these infections and these other triggers can activate the peripheral immune system. And these then have downstream consequences that can influence immune-brain communication, sometimes acutely and other times more chronically. And it's the more chronic activation of your immune system that sometimes might be a target for some sort of an intervention, if in fact it's one of the factors that allow sort of negative symptoms to persist. And we can measure aspects of an individual's peripheral immune system from a blood sample, and we'll talk a little bit about that. Some of the benefits for looking at the immune system in brain health and in neurodevelopmental disorders. So we think of these in the context of biomarkers and how biomarkers of individual differences in clinical presentation, it might be helpful both in treatment of the individuals, but also in understanding some of the biological mechanisms that might be contributing to the clinical presentation. So if we can, in fact, identify how the immune system is different in different individuals in both typically developing and in neurodevelopmental disorders, we might be able to subgroup individuals based on their shared biology, and you might have already heard a little bit about that this morning. We can provide new biological targets for the development of new drugs or therapies. And an important aspect of this is also to understand who might benefit from those therapies. So there's a lot of work going on around the world that's expanding the concept of diet or prebiotics, probiotics, live biotherapeuticals as intervention strategies that are more holistic in their approach to neurodevelopment. But of course, we need to understand who will benefit from those and what are the signaling systems that they're targeting. But in the long run, what we want to do is we want to use these precision health approaches to match individuals to the best treatment approach that's going to work for them in the shortest timeline. So in the Pond Immune platform, we collect a series of biospecimens. We're also now collecting stool moving forward, but we collect a sample just to get your basic clinical lab white blood differentials. So this is a standard lab test that often people do just to take a look at the proportion of the blood cells in an individual's circulating system. We collect plasma, and we collect what's referred to as peripheral blood mononuclear cells. So these are those T cells, B cells, NK cells, and monocytes that are circulating in your blood. And then we can also collect serum where we can measure both immune and endocrine markers. So this is the panels that we use. And I'm just going to tell you a little bit about the research that we have been doing using these. And so this is just a schematic to remind you that the immune system is, again, it's embedded in tissues all over your body. In this case, we're looking at the barrier of the gastrointestinal tract. So you see those microbes that are lining the mucosal barrier. And then we have these epithelial cells, but right on the inside of there is your mucosal immune system, which is very rich in these different immune cells. And that purple immune cell that has its extension into the gut is a dendritic cell. It's actually one of the first responders that can sample the metabolites or the gut and actually then communicate with other immune cells or travel through the bloodstream. But this mucosal system lies right under that enteric nervous system. And this is that system of neurons that wraps your gastrointestinal tract that's very important for motility, but also sends signal up through the spinal cord to the brain. So you can see this complex system at the barrier right at the bottom is a powerhouse of information of which this peripheral information can signal your brain. And in the blood, what you see in the little schematic that's there is actually these immune cells, whether they're from the mucosal system or they're from other peripheral systems, and the plasma contain both these immune molecules that are produced. They contain those cells, which we can collect at the bottom. Those are those PBMCs that I spoke about in the previous slide. Or we can take that plasma, and in the plasma, we can look not only at immune proteins, but we can look at gut inflammatory proteins, or we could look at host metabolites. And all of these allow us to build what we refer to as a biological signature that tells us something about the individual's biology. And so I'm just going to give you one example of how this might be applied. And this is work from a graduate student, Shane Cleary, in my lab, who's just finishing up his PhD. And through the PON network, he was able to take a look at the behavioral profiles of 1,800 participants that were enrolled, that included people who were, or individuals that were typically developing either a diagnosis of ASD or a diagnosis of ADHD, and using these comprehensive behavioral characteristics, he was able to identify, using some clustering tools, 6 different clusters of individuals, and these individuals had very different profiles. So there's 2 things that we would take home from this. First of all, cluster 1 is mostly typically developing individuals, but it also has a mix of ADHD and ASD, and these individuals really have very low behavioral deficits with respect to social behavior, repetitive behaviors, or anxiety, externalizing, internalizing behavior, or hyperreactivity or inhibition. Whereas this cluster 2 is a group of individuals that was 80% ASD individuals and 20% ADHD that had a really high burden of symptoms across all of these domains. And so here, when we look at these, I'm quite sure that Evdokia has already talked to you about the fact that these diagnostic labels always don't always line up with the clinical phenotype. And so by looking at and profiling individuals across these domains and identifying these clusters, we then can take a look closer to see, is there some biology that distinguishes between these 2 clusters? And that's the sort of work that we're doing right now, using the immune samples that a subset of these individuals provided the immune platform. And so this is just one example of how that would look. And so in some pilot work that we did in order to get some more funding to do a bit more research, we looked at cluster 3 and cluster 4. And these are interesting. So if you are looking closely at all those bars at the top, I can debunk it for you. So cluster 3 had a profile that had normal social communication, normal repetitive behaviors, but high levels of anxiety, internalizing behavior, and externalizing behavior, whereas cluster 4 had more of a ASD-like phenotype, which had deficits in social behavior and repetitive behaviors, but somewhat normal levels of anxiety, internalizing and externalizing behavior. And Chow, who's visualized here down the bottom, actually did a comparison in a small group of people using what's called single-cell RNA-seq, where she can actually map not only the immune cells in those individuals, but their immune program. And what she identified was there actually was an immune phenotype that we think that might explain the differences in social behaviors in these two groups of individuals. And so this is an observation that we're hoping to follow up on it was just a pilot to take a look at, but it just sort of gives you an idea of the tools we can use and the precision we can get to actually start mapping individual immune phenotypes. And this is a picture of what this might be used. It's not exactly the outcomes that we're looking at right at the moment. It's one that I created for a different overview, but you can imagine that if you have a series of individuals, whether they be typically developing or neurodevelopmental disorders, and you can map them based on their biology into subgroups, in the long run, you might be able to, in this schematic, I've identified 8 different biologically divided subgroups that's based, first of all, on an MRI, a look at their microbiome, and then a look at these PBMCs. And so that you could map people to these individual subtypes, and then potentially we can use those subgroups in clinical trials and other ways to determine if you have this subgroup, what's the best treatment approach for your for that individual. So I'll end it there and be happy to answer any questions that might come up. I can't see anybody, so I assume.
That's okay, now we're here. So for Dr. Foster, remember to do your slide. So somebody has a slide. You want to go for it?
So the clinical setting, I would love to practice this kind of thing. No one seems to be aware the immune
Abdikia, can you repeat the question? I can't actually hear.
Yeah, I will repeat it after. Let me just get it.
So the question is regarding clinical use. In my experience, my own experience, there's a lot of research interest in inflammation and everything that has to do with immune, but I don't see anybody. And it's just painful to watch your child grow and there's knowledge that is just being. Can this be done privately? Can this be?
Okay, so I'll get the question to her and then I'll have a follow-up to whatever her answer is. So Jane, the question is very interested in looking at immune differences and their behavioral correlates in mental health and neurodevelopment. We talk about research a lot, but there is no clinical testing that people can access that is informative right now and help guide treatment. So I think the question what's the nature of the lack of clinical testing? Is it that we're not ready? Is it that we just don't have it, but we can actually in the publicly funded system, but it can be accessed other ways? Do you have recommendations or thoughts around the lack of clinical testing in immune?
So I think that and I think probably Abdikia, you could speak to it as well from a clinical perspective, but part of the reason I showed that I selected those examples I did is that this is very recent work that bioinformatic tools that we now have are allowing us to cluster individuals in larger data sets to provide the framework to test in smaller data sets. So in the example that I showed you, we got these behavioral clusters from 1,800 people, but in the immune platform, we did the testing in a small number of people. So I think that we're not, the approach to biomarkers is coming of age in behavioral disorders and in psychiatry, and we're lagging behind some of the other biomedical specialties that had a blood signal early on. And so I think that clinicians and Abdikia, you could really expand on that. Clinicians in looking at behavioral disorders in psychology and psychiatry are opening up to thinking about biological subgroups. And I think that there has been some evidence of some imaging studies that have supported that as well. But I think it's correct. We're not yet using anything in a standard way in the clinic. But we have to first discover it, then validate it, and then test it. So I think we're getting close on those levels.
Yeah, so I'm just going to quickly follow up. It's really important that we understand the difference between a research experiment and a clinical test. By the time we get to a clinical test, we need to know how accurate it is in predicting something that is of use clinically, of clinical use, when it's behaving appropriately and when it's not behaving and should not be used. And we should have pretty clear ideas about what we call positive predictive and negative predictive value. How often does it get it wrong and predicts that you should do something, and how often gets it wrong and predicts that you shouldn't do something? And I would say we're not there in this space.
Correct.
Although we are truly, truly committed to the space. If it makes you feel any better, we're going to talk about it later, but we are going to collect immune samples that are appropriate for immune analysis for all the kids in this new phase. And we're going to collect stool and urine to look at how, and saliva, to look at how other markers that map onto the immune system may be helpful in clinical practice downstream. But right now, if we order the test, we will know what to do with the results, basically. That's what it comes down to, right? Who's next?
Have you had anything, I guess I'll give you our but I think like my son, he has autism, ADHD, and anxiety. He basically didn't have an immune system. He never learned to blow his nose for himself. Had a strep throat, he didn't have like 9 times. So it's like he didn't have. He had RSV as a baby, pregnant, so can all that be?
So can I summarize for her? Okay, this is not kidding the question. So example of a young man who's now a young adult, has autism, ADHD, anxiety, seemed to not feel it when he had infectious as a young child. So people by randomly identifying that he had infectious conditions, but he experienced no particular distress around them. And remind me, the next step now is.
Oh, and then I have.
Oh, and then my family has history of potentially autoimmune related phenomena from the description of mom. And so the question is, do you think there is a link to this kind of profile? And do you have any thoughts about next steps?
So remembering that I'm a PhD neuroscientist, I did not go to medical school, which is a caveat that I think is important to note. There is a fair amount of evidence in animal models and in clinical populations of interactions between exposure to infections early in life, whether it be maternally or in early life, that can actually change the way that the brain is wired. And the reason that is because of those first few slides I showed, where your gut bacteria are talking to your young immune system, when you're first born, and that crosstalk really does not only set a tone for your immune system, but it influences brain wiring. And so there is evidence of potentially these interactions that I'm talking about that are part of normal, healthy development in interacting with your host genetics, that is the DNA you're born with, because the DNA you're born with really provides us with our risk factors at some level. So, when we think about, when I think about the microbiome and the immune system, I see it as the environmental components of what we call gene-environment interactions that might contribute to something downstream. And the place where there's a lot of evidence of that actually mechanistically being important is in some types of allergy where kids are exposed to antibiotics early in life for a whole host of different reasons, which then messes up the way that the maturation and the development of their microbiome, which in the end does normalize. But one of the downstream consequences sometimes is an increase in asthma and other allergies in those kids. So there is evidence from epidemiological and scientific level of these associations. What we're not at yet, as Evdokia said in the previous question, is an ability to point to an individual and find out what are the mechanisms underlying that individual. And that's why I like that sort of staggered approach of using multiple domains to try to think about the biology, not just the immune system, not just the gut, or not just the brain.
Yeah, and I guess I could just, for the specific last part that you mentioned, we do know, so epidemiologically, in neurodevelopmental conditions, we see in family members a higher rate of autoimmune conditions. So there is likely some story there to be told, although we don't quite know it. And then with Steve in the room, I think at some points when the genetic work happens, genes that are relevant to the immune system have come up into some of the neurodevelopmental genes. So this is a story that's really interesting, but exactly that we do not know how to map it. That's why we're committing resources for the renewal also to clarify the story a bit better. I see Alana standing, which makes me think, okay, so I see two, and I'm just gonna be, nobody else presses their mic. So get Peter, go.
Okay, this is completely favorite. He's always really sick because it starts to live like cases.
So the question is about a young man who, when he has fever, he's actually doing better in terms of social and cognition. You have a quick response, Jane, because we're gonna be quick now.
Yeah, I think that there are anecdotal stories of that. I think mechanistically we don't quite understand, but there are also anecdotal stories in pregnancy of people whose allergies disappear. I think it points to the point that the part of your brain that responds to infection is the part of your brain that responds to stress, and it's the part of your brain that also responds to food. So our brain in itself, where we manufactured the hypothalamus and the extra hypothalamic circuits, the way that connects to other parts of your brain could influence cognition and other things. So it just suggests that the immune system is playing a role, but I don't think it tells us anything mechanistically.
Yeah, I mean, I'm gonna add the caveat to this. So first of all, the stories are there for sure. There is a large study that was published last year, but overall did not find an association. So it's probably a subgroup. But also remember that fever on its own has a mechanism. So just because we have fever when we have an infection, it doesn't mean that it's the infection that may be responsible for the phenomenon. It could be the fever themselves, what happens to the brain when we develop a fever, and there's a whole model proposed outside of the immune system that could account for the phenomenon. So again, we have to figure out the mechanisms. Could be immune, it could be something else. So last question over there, and then we have our talk.
I was just wondering, are there recommendations coming out of the, early exposure to antibiotics, things like help prevent later issues with the brain, and also connect with the other way around, allergies early in life, put that into more
Okay, so this is fundamental immunology, so I'm gonna go to you, Jay. So the second part is, if you have allergies, like could the direction be different? So if you start with allergies early on in life, could they themselves be causative to immune differences later on? And the first part was, are there any recommendations that come from what we are learning from early antibiotic use and later sequela in terms of clinical care?
So so on the antibiotic side, the less antibiotics is good is the evidence across the board. There is some evidence of these new short-term antibiotics, and maybe you know something more about it, Evdokia, that are, that you only have to take a very short course, that they have less of an impact on the microbiome and the related immune response to that. And
allergy
Oh, I don't know of any evidence that allergy directly has a causative link to later risk related to neurodevelopmental disorders, but I do think it probably reflects a genetic risk of immune disorders in general.
Yes.
I think that's probably where it might line up if we were looking. And in the immune platform, we do look at allergy and other things, and there is a slight increased incidence in allergy and other immune conditions in neurodevelopmental disorders in our 600 people we've looked at compared to typically developing, but that's just a self-report look, not any mechanistic look. It doesn't seem to change some of the signaling cascades that we're looking at in the cells.
So again, right? So allergy common but higher in our population. Again, part of the story, but we don't know the exact story. And the less antibiotic, the better. And there's a lot of effort to try to figure out minimum doses, absolute necessary conditions. It's not just for us; it's for everything. Like we're in a crisis of microbiome.
Yeah, sometimes the reason for the antibiotic is more damaging than the antibiotic.
We're trying to minimize antibiotic use anyway. We can talk more about cascades for pharmacology at the pharmacology workshop. So I know somebody's waiting to take you to your genetics tour for your lunch. I'm gonna pass it on to Alana. We're gonna take our lunch break. You're gonna go for genetics. You're gonna come back and we're gonna change the order of the afternoon so you get to actually hear from everyone. Thank you, Jane. Yeah.
When you first walked in, there was a little registration table. If you'd like to take a tour, Elim, Andrada.
Thanks,
and
guys. Bye.
Sheer will be there.